Okay, I'm Zhenkun Lu from Mayo Clinic. It's my great pleasure to introduce tonight's keynote speaker, Dr. Li Zhou. So Li got his bachelor degree from Sun Yat-sen University in 1992. Then he went to United States and trained under Dr. Bruce Stillman at Cold Harbor and got his PhD degree. Then he did a postdoc training at Dr. Steve Adler's lab. In 2004, he got his independent position at Harvard University, and he rising quickly and become a full professor in 2013. And he is now also the James and Patricia Poitras Endowed Chair and co-leader of Cancer Cell Biology program in Dana-Farber Cancer Institute. He also got many awards such as V Scholar Award, ACS Research Scholar, Leukemia Lymphoma Society Scholar. So Li is really a leader in the study of the RPA ATR signaling pathway. So if you read many landmark papers in this pathway, you're gonna come across Li's name many times. So when he was in Steve Adler's lab, he was one to find that APR is really a sensor for single-strand damage that help to recruit ATR and help ATR activation. So after he established his own laboratory, he did many magnetic study really show step by step how different protein get interacted with each other, how different post-translational modifications such as phosphorylation, ubiquitination, accumulation can coordinate to help ATR activation. So his work really helped establish current paradigm how ATR is activated. So another big contribution of him is actually he really expanded how the ATR pathway works, and it's not just only a checkpoint protein. He showed that RPA really can sense the R loop. Also, recently, in a really groundbreaking paper published in Science, he showed that ATR actually have a mitosis-specific function regulating chromosome segregation. So also, he did venture into the translational study and investigating how ATR inhibitor can be used to selectively kill cancer cells. And he have a very exciting paper showing that for the cancer cells have this alternative lengthening of telomere, they are hypersensitive to ATR inhibitor. So really, Li did many very unique and groundbreaking work in this RPA ATR pathway, and really a leader in this field. So without further ado, I want to give the podium to Li.
Is it on? Thank you, Zhenkun, for your kind introduction, and thank you, organizers, for the opportunity to come to this meeting. As Zhenkun mentioned, many years ago, I was a graduate student in Dr. Bruce Stillman's lab at Cold Spring Harbor. Coming back for a Cold Spring Harbor meetings really gives me a feeling of homecoming. I'm sure Dr. Diffley will say the same. So today, I would like to share with you some of the recent projects in my lab. So one of the major research interests in my lab is to understand the genomic instability in cancer cell and find ways to target this particular vulnerability in cancer cell. As I'm sure you're all very familiar with this figure from the classic review article published by Doug Hanahan and Bob Weinberg several years ago. So in this paper, they tried to summarize the hallmarks of cancer. And they pointed out that genome instability and mutation is one of the enabling characteristics of cancer, meaning that this is one of the driving forces of tumorigenesis. So we know that this is not just a hallmark, it actually is a vulnerability of cancer cells. So it is known for many years now, you can actually target cancer cells by introducing DNA damage using radiation and chemotherapeutic drugs. But now we're in the age of targeted therapy. So our goal is to have a deeper understanding of this vulnerability in cancer cells and find ways to selectively target this vulnerability. So my lab, as you know, has been interested in DNA damage sensing and signaling for a long time. And the signaling pathway that respond to DNA damage is controlled by these two master kinases, ATR, ATM. So this pathway can respond to different type of DNA damage, such as DNA double-strand break, stalled DNA replication forks, and this pathway eventually can control many cellular processes, such as cell cycle progression, DNA repair, DNA replication, and so on. So this is an important pathway that keeps the genome stable. And my lab is particularly interested in the ATR pathway because ATR pathway not only respond to double-strand DNA breaks, but also to different type of DNA replication problems, replication stress. And this is particularly relevant to what happens in cancer cells because we now know that many oncogenic events could lead to replication stress, and also many therapeutic drugs can also elicit replication stress response. So therefore, a better understanding of the replication stress response mediated by ATR will give us a better idea of what happens in cancer cell and how they respond to therapeutics. So many labs, including mine, have worked many years trying to understand how ATR is activated. We now know that at least one way to activate ATR is through single-stranded DNA. So single-stranded DNA could be generated at double-strand breaks through this process called resection through exonuclease activity. Single-strand DNA could also be induced as stall replication forks through the uncoupling of different components of replication forks or through nucleolytic processing of replication forks. So once single-strand DNA is generated at breaks or replication forks, they will be recognized by this protein RPA. RPA is a high-affinity single-strand DNA binding protein that recognizes single-strand DNA in a non-sequence specific manner. So once RPA codes single-stranded DNA, this RPA-coded single-strand DNA will function as a platform to recruit the Atr kinase, its activator proteins, including RAS17, 911 complex, TOP-BP1, and ETAA1. So all these proteins, these activators, regulators of ATR, will be recruited with the kinase onto RPA-coded single-strand DNA. And perhaps RPA-coded single-strand DNA can also recruit some of the substrates of ATR. So this is a platform that can enrich the kinase, the regulators, and some of the substrates at the same place, at the same time, to facilitate the function of ATR to phosphorylate the substrates. And perhaps this is a mechanism that could activate ATR at the right place at the right time. Okay, so this is perhaps a model to explain how ATR pathway is activated. But today, I would like to talk about some other questions that we're interested in. So first, we're really interested in how genome instability arises in cancer cells and how it is sensed, because this is really perhaps a major trigger of ATR in cancer cells. And we're also interested in how ATR suppresses genome instability. So we know that ATR is perhaps a barrier against genome instability in cancer cells. And finally, we would like to understand in what situations targeting ATR may be beneficial for cancer therapy. So this brings us back to this important question. So is ATR checkpoint a good target for cancer therapy? Because I just told you that ATR is a very important protein. It keeps the genome stable. So why would you target such an important protein in therapy? The thinking here is actually very simple. So during the process of tumorigenesis, as you know, the genome in cancer cells will become more and more unstable. And then at some point, the genome instability within a cancer cell can activate the ATR checkpoint. And under this condition, in this context, ATR checkpoint is actually protecting cancer cells. is helping cancer cells to cope with the instability within themselves. And in this situation, if you take away ATR checkpoint, you're removing a protective mechanism. And now the instability in cancer cell is enough to let the cancer cell to commit suicide, to kill themselves. So therefore, ATR under oncogenic stress, under replication stress, is actually a protective mechanism in cancer cells. By inhibiting ATR, you might be able to selectively kill cancer cells. So coming back to this question, so is ATR checkpoint a good target for cancer therapy? So my guess is that ATR inhibition might be beneficial for, to therapy in specific context. So you have to find that specific context. You have to find a cancer cell that can commit suicide without ATR. So this is actually one of the major efforts we are making now, try to identify the intrinsic problem, intrinsic vulnerability in cancer cell that can make them addicted to ATR. Okay, so question one. So what are the causes of genome instability in cancer cells? Okay, so you all know that the genome instability in cancer cell could arise from many sources. Some are intrinsic, some are extrinsic. Extrinsic sources such as radiation, carcinogen, and intrinsic sources, they could be normal cellular processes such as DNA replication, mitosis, telomere shortening, and under pathological conditions, for example, in cancer cells, some of these intrinsic problems can get worse. So both intrinsic, extrinsic problems contribute to instability. So today, again, I would like to draw your attention to one particular type of instability arising from transcription and RNA biogenesis defects. Okay. So, as Dr. Fogliani just mentioned, so during transcription, normally, when RNA is synthesized by RNA polymerase, it comes out from RNA polymerase, and it will be processed by RNA processing factors, such as RNA splicing factors. So these processes occur in a transcription-coupled manner. So when this whole thing happens very smoothly, typically RNA will not form stable hybrids with DNA. However, this could be quite different if something is not so smooth during transcription. For example, when RNA biogenesis is defective, or when topological problems could not be resolved during transcription elongation. So under these conditions, the RNA generated by RNA polymerase could now form a stable hybrid with a DNA template to form this DNA-RNA hybrid, and one of the DNA strand will be displaced as single-stranded DNA. And this structure, people call them R loops. So R loops actually have some normal functions, positive functions. So R loops have been involved in transcription control, class switch recombination, telomere protection, and DNA repair. But R loops could also be a problem. When the level of R loops gets too high in the genome, R loops could interfere with transcription, interfere with RNA biogenesis, and it becomes a source of genomic instability. The reason why R loops could be a source of genome instability is perhaps the collision between DNA replication forks and R loop is a source of DNA breaks. And also, R loops could be recognized by some of the structure-specific nucleases because their structural similarities with DNA repair intermediates. So this is perhaps the reason why cells couldn't tolerate too much R loop. So therefore, the levels of R loop has to be properly controlled. So we are curious how cells can control R loop levels. So there are two ways cells could control R loop levels. One is through efficient RNA biogenesis, RNA processing in a transcription-coupled manner. And the second, there's a group of proteins, including helicases such as synataxin that can unwind a DNA-RNA hybrid, or RNAs, such as RNase H, that can specifically degrade the RNA in RNA-DNA hybrid. So when you think about how these proteins can function to suppress R loop, so for this group of protein, it's actually relatively easy to understand because many of these proteins, they travel with the transcription elongation machinery. So they're at the right place at the right time to suppress R loop formation. And for this second group of protein, right, so they're more like DNA repair proteins. They have the activity to remove R loop, but it's not entirely clear how they recognize R loop in cells. So this is actually one of the questions we had several years ago. So we stumbled on this question because we did a proteomic screen using RPA-coded single-strand DNA, try to identify proteins that may regulate the Atr pathway. So through that proteomic screen, we found quite a few proteins that are functional, important in the Atr pathway. And in addition to these proteins, we also found a protein called RNase H1. So this was quite a surprise to us at the time because it suggests that perhaps RPA is interacting with RNase H, and this could implicate RPA in the control of R loop. So as I mentioned, in the R loop, in addition to DNA-RNA hybrid, there's a displaced single-stranded DNA. And my postdoc, Hai Deng, started to wonder if RPA is binding to that displaced single-strand DNA in the R loop. So he confirmed the interaction between RPA and RNase H. The physical interaction is definitely there. And then he modified a protocol to use a monoclonal antibody called S9.6 to stain for regions in the nucleus that is enriched for R loops. He could actually detect nuclear foci of R loops. So these are possibly the regions in the genome that are enriched of stable R loops. So if we knock down one of the DNA-RNA helices, the Aquarius, we can see that the levels of R loops was increased. And if we express active RNase H1, we can remove R loop, that staining goes down. So this is a control experiment telling us that our staining is specific. So here we start to look at if RPA actually localized to R loop. So here we're looking at R loop staining. So this is the catalytically inactive RNase H1. They colocalize very well, telling us that R loop and RNase H1 colocalize with each other. So here we're looking at RNase H1 and RPA. We can see them colocalizing. RNase H1 and RPA colocalize. So here we're looking at R loops and RPA. Again, they colocalize substantially. So now we know that R loop, RNase H1, and RPA, they all colocalize with each other in cells. So we also know that this colocalization between RPA and R loop is dependent on the hybrid. So if we express RNase H1 to remove the hybrid, we lost R loop staining, we also lost RPA staining. So this result suggests that perhaps RPA is present at R loops in cells together with RNase H1. And I want to point out that we could detect RPA at R loop even in the absence of DNA damage signals, and even in cells that are not undergoing replication. So we think that R loop is probably directly sensing R loops. RPA is directly sensing R loops in this situation. So how does it work if it's there? RPA may stimulate the activity of RNase H1, so this is one of the possibilities. So we tested this idea. Tribble in my lab, he purified RPA complex, wild type, and the catalytic mutant of RNase H1. So we use this in vitro assay to measure the activity of RNase H1 to degrade RNA in a DNA-RNA hybrid. You can see that when we titrated in RPA, RPA greatly stimulated the activity of RNase H1. And we can also see this on R loop-like structure. And again, when we titrated in RPA, RPA greatly stimulates the activity of RNase H1 on this substrate. So this is telling us that, yes, indeed, RPA is helping RNase H1 to cleave the hybrid more effectively. The second possibility by which RPA could regulate RNase H1 is to recruit RNase H1 to R loop. To test this, we try to make a mutant of RNase H1 that doesn't bind RPA. So we first map the region that binds to RPA in RNase H1. It turns out to be the N terminus, and that is the DNA RNA hybrid binding domain in the protein. And very fortunate to us, Dr. Wei Yang's lab has solved the crystal structure of this part of RNase H1. They found that next to the region that binds to DNA RNA hybrid, there's a conserved region called basic ridge. So this region is not directly involved in binding to DNA RNA hybrid, but it somehow is there and is conserved. So we wonder if that region is actually binding to RPA, and we introduced point mutations in that region, and you can see that point mutations in this region can disrupt the binding between RNase H1 and RPA. So now we have a point mutant that specifically disrupts the interaction between RNase H1 and RPA. So when we expressed this mutant in cells, you can see that the catalytic inactive RNase H1 can co-localize with R loop quite well, but the RNase H mutant lacking RPA binding can no longer localize to R loop. So yeah, RNase H1 really needs that interaction to localize to R loop. And also, functionally, that interaction is also important because when we express wild-type RNase H1, we can remove R loop and remove RPA staining at R loop. But when we express the point mutant that is unable to bind RPA, so this point mutant can no longer remove R loop and remove RPA from cells. It's functionally important. And this interaction is also important for the ability of RNase H1 to suppress R loop-associated genomic instability. So here, if we knock down Aquarius, so that helicase that suppresses R loop, we can see an increase of CAP1 phosphorylation. So CAP1 is the substrate of ATN, indicating that genomic instability is increased when R loops goes up. We can suppress that by expressing active RNase H1. So we remove R loop, we remove instability. But when we express that point mutant of RNase H1 that couldn't bind RPA, this mutant failed to suppress R loop-associated genomic instability. So again, this is telling us that that interaction between RNase H1 and RPA is critical for the function of this protein. So, this result suggests to us that RPA is perhaps a sensor of R loop. It has at least two important functions in regulating RNase H1. One is to bring RNase H1 to R loops, and second is to stimulate the activity of RNase H1 at R loop to help it to function more effectively. So one interesting point out of this part of the project is that we now realize that RPA-coded single-strand DNA is present at R loops. So this, at the time, really raised a new question to us. That is, does ATR respond to aberrant R loop accumulation? Because we know for a while, RPA-coded single-strand DNA is one of the signals that trigger ATR activation. And we will also want to ask another question, that is aberrant R loop accumulation relevant to cancer? Yeah, and if so, can it be targeted therapeutically? So these are two relevant questions together. So coming back to this model, how R loops could arise from different type of problems. And one type of problem is in our mRNA biogenesis. So yeah, many years ago, people know that if you deplete certain splicing factor, you can induce R loops. So the question we would like to ask here is, can you find this situation in cancer cells? Can you find cancer cells with splicing defects? So fortunately, I have a colleague in my cancer center, Dr. Tim Graubert. So Tim studies blood cancer, including different type of leukemia. So several years ago, he and his colleague found that one splicing factor called U2F1 is frequently mutated in myelodysplastic syndrome, MDS, and acute myeloid leukemia, AML. So this two kind of oncogenic cancer situation indeed associate with splicing factor mutations. Now, yes, after several years, it is actually quite surprising that splicing factor mutations are very prevalent in MDS and AML. Almost 50% of these patients have splicing factor mutations. And interestingly, splicing factor these mutations, they're always hotspot mutations, missense mutations. You never see nonsense mutations or frame strand mutations. They're hotspot mutations. And they're always heterozygous mutations. So people thought that these mutations are likely gain-of-function mutations because how they accumulate in cancer cells. So the question we ask is, do these mutations affect R-loop levels? So we pick one of the most prevalent mutations in MDS and AML, that is in U2F1, to test this idea. So because we thought that this is a gain-of-function mutant, so we established cell lines using different cancer cell lines, including HeLa cells and leukemia cell lines. So we try to express wild-type U2F1 or U2F1 S34F, the very prevalent mutant in cancer. We express them at very low level, even below the endogenous protein, and try to recapitulate their gain of function. So what we found is that when we express the mutant, this mutant can actually induce R-loop potently, but not the wild-type protein. So we know that we're looking at R-loop because when we express RNase H1, we can actually suppress that signal. So we're confident we're looking at the right thing. And as I mentioned, when we induce R-loop, we can actually induce RPA phosphorylation. And here, we also found that when we express that mutant splicing factor, we induce RPA phosphorylation. And again, it could be suppressed by RNase H1. So it is indeed coming from R-loop. And furthermore, we found that R-loop-induced RPA phosphorylation is ATR dependent. Yeah, if we treat cells with ATR inhibitor, cells expressing that splicing factor mutation, we can eliminate RPA phosphorylation, but ATM inhibitor did not do the same thing. So together, these results is telling us that that particular splicing factor mutant can induce R-loop, and R-loop perhaps is activating the ATR pathway. So here, we can ask the next question, is ATR actually important? Is it functionally important in this situation? So now we're looking at cells expressing wild-type splicing factor or that mutant, and we treat them with either ATM or ATR inhibitor. You can see that the cells expressing the mutant, they generate much more gamma-H2X when we treat them with ATR inhibitor. And also, the cell lines expressing wild-type protein wild-type protein and the mutant protein, they are equally sensitive to ATM inhibitor, but the cell line expressing the mutant protein is more sensitive to ATR inhibitor. So this is a very specific sensitivity to ATR inhibitor, but not ATM inhibitor. And it is specific to the mutant form. So now we wonder, right? So we tested one mutant. Is this a very unique situation? So now we tested a panel of U2F1 mutations that people found in MDS and AML, and turned out that every single one can make cells more sensitive to ATR inhibitor. So this is a very general feature of this kind of mutant, at least in this splicing factor. Okay, so now we know, so ATR is actually important. ATR is helping to keep these cells alive if they express this kind of splicing factor mutations. So now we would like to ask another question. Can we actually enhance R-loop accumulation in this situation? Perhaps this is a way we can sensitize this kind of cells to ATR inhibitor. So there's a way to enhance R-loop, to interfere with splicing in cancer cell. So people have developed splicing factor inhibitors. So the idea here is that in normal cells, splicing requires multiple factors, A and B. You have very efficient splicing. In some cancer cells, they already have mutation in a splicing factor. So now splicing is already partially compromised, but they manage to survive. So the idea here is that if you can develop an inhibitor of a splicing factor, you can actually further compromise splicing, and this could create a synthetic lethality based on the splicing defect and selectively kill cells with splicing defect. So here, we want to use those splicing inhibitors to see if we can induce R-loop. So here you can see one of the splicing inhibitors can induce R-loop, E7107. Another splicing inhibitor, Plant B, can also induce R-loop. And we know that we're looking at R-loop because it can be removed by RNase H1. So here, we are looking at a cell population. We use the splicing factor to induce R-loop. And we also see RPA, phospho-RPA signal is induced. You can see them, they go out together. So these are two associated features induced by the splicing factor. When we express RNase H1, we can suppress R-loop and suppress RPA phosphorylation. So now we have a way to induce R-loop using splicing factor mutations. So the question is, can we use splicing factor inhibitor to sensitize those cancer cells to ATR inhibitor? So here, in cells expressing splicing factor mutant, R-loop level is already up. If we treat them with splicing inhibitor, R-loop level goes further up. So you can further increase R-loop level. So here you can look at RPA phosphorylation, it's the same thing. The mutant itself increase R-loop. Mutant plus splicing factor inhibitor, it goes further up. And that could be suppressed by RNase H1. So here we're looking at DNA damage, gamma H2X, in cells expressing the mutant, either splicing factor inhibitor or ATR inhibitor can induce DNA damage, but a combination of the two can induce much more DNA damage. Here you're seeing the same thing. The combination of ATR inhibitor and splicing inhibitor induce a lot of DNA damage, and you can suppress that by RNase H1. So this kind of DNA damage arises from R-loops. Okay. So here you're looking at cell death, apoptosis, and again, cells expressing the mutant, you see a clear increase of apoptosis when we combine ATR inhibitor and the splicing factor inhibitor. And here we're looking at cell death, and again, only the cells expressing the splicing factor mutant are sensitive to the combination of ATR inhibitor and splicing factor inhibitor. Okay, so now we know that we can sensitize cells to ATR inhibitor by using splicing factor inhibitors. Okay, so far I have shown you that in model cell lines, in cancer cell lines where we try to express this splicing factor mutation, this splicing factor mutant can render cells sensitive to ATR inhibitor, and then we can sensitize them using splicing factor inhibitors. But what about the relevant cell line? Because these are not the real cells in which this kind of mutation are generated. So people know that in NDS and AML, this kind of splicing mutations occur very early during the pathological path. And it normally accumulates in progenitor cells, hematopoietic progenitor cells. To test this possibility, we actually sort CD34 positive human progenitor cells, hematopoietic progenitor cells, and we express either the wild-type protein or the mutant protein in them using lentivirus. And you can see that expressing the mutant protein in progenitor cells increased DNA damage, and also increased ATR inhibitor-induced DNA damage and ATR inhibitor-induced cell death. So now we know that even in the relevant cell line, cell type in progenitor cells, this kind of splicing factor mutation can indeed increase DNA damage, increase the sensitivity to ATR inhibitor. Okay, So this is kind of a simple model trying to explain what we're seeing here. We know that in MDS cells that express the U2F1 mutant. So there are two major consequences from this mutant. So people know that this kind of splicing factor mutations could promote RNA alternative splicing of a subset of genes. So now we show that this kind of splicing factor mutation could also promote accumulation of R loops. And both of these changes could contribute to tumorigenesis. But the mechanism is still unclear at the moment, I have to say. People are actively working to understand how alternative splicing or R loops could perhaps contribute to tumorigenesis in this setting. We also want to suggest that the accumulation of R loops actually could lead to accumulation of DNA damage, eventually cell death in this situation. But this normally doesn't happen very efficiently because R loop also activates ATR and triggers a feedback loop to prevent cells going down this path. But when you inhibit ATR using inhibitor, so now you're removing this feedback loop protective mechanism, and now this pathway becomes much more efficient. So cells are dying in a more specific way. And we also know that using splicing factor inhibitors such as E7107 and PLAT-B, you can induce R loop and sensitize cells to ATR inhibitor. So we would like to suggest that the combination of ATR inhibitor and splicing inhibitor perhaps is an effective and selective way to kill cancer cells with this type of splicing factor mutant. So the big question we're facing now is how general this phenomena is. So we tested a limited number of splicing factor mutations so far, in our hands, splicing factor mutations, they do increase R loop and make cells sensitive to ATR inhibitor. But we still don't know if this is a general thing in cancer cells. Very recently, a paper reported that many splicing factors are actually mutated in a broad spectrum of cancer types. So splicing factor mutation is actually quite a general type of mutation in cancer cells. So the challenge for us for the next phase is to find out which type of splicing factor mutations could generate R loops, and if we could target those cancer cells with ATR inhibitors and splicing factor inhibitors. So at the end, I would like to thank people who work on this. Hai Deng is the postdoc who initiated both projects, and Tribble is the biochemist in the lab who helped him with the in vitro assays. And thank you for your attention. Happy to answer your questions. Oh, yes.
Oh, hi. This is a really nice talk. I had two quick questions. So one is regarding the role of ATR at the R loop. I mean, is it akin to its role at the replication fork? I mean, does it directly prevent breakage of the R loop, or could it be also involved in the recruitment of RNase H1 via phosphorylation of RPA, for example?
Right. So that's something we're actively working on now. So we don't know all the details yet, but I think ATR has a role in preventing this kind of break formation after the collision of replication fork and R loop. That's perhaps where ATR functions.
Sure. And a very quick general question. So is there any way of figuring out if R loops are enriched at, let's say, the early replicating fragile sites, now that you have a marker for looking at R loops?
Right. So we haven't looked at that very deeply yet, but that's a very interesting question, yes.
Thanks.
Yeah. Mike, yeah. Oh, is it on?
So a couple of quick questions.
Sure.
So first of all, when you generate an hybrid and you don't generate RPA filaments and a non-transcribed strand, you might form a secondary structure like cruciforms, okay?
Right.
So in this view, RPA filaments would protect and prevent formation of cruciforms, okay? So I wonder whether in the pathological situation of splicing mutants, you deplete, let's say you have too many hybrids and not enough RPA to prevent that kind of phenomenon.
That's a great model. We don't know yet, but I like your idea, yeah.
Second question is G1 versus S. So my expectation would be that you would have more R loops in G1 than in S phase because the fork can proceed also across the hybrid without any problem by uncoupling leading and lagging strand. Do you see any difference in sensing in G1 versus S phase?
Yeah, there is some interesting. So in S phase, of course, you have this collision problem, right? So we could see R loop. We see RPA and R loop in G1 without replication. So that's why we think, yeah, you don't really need replication to see it. But in S phase, you do see an increase. So we think that the collision probably is doing something too to bring in more RPA. Yes. Yes.
Very nice talk. A related question to two previous ones. So, do you check whether you observed RPA phosphorylation only when the cell entered X phase, or the replication?
Yeah, we can see in the G1. Yeah.
You can also, even for the phosphorylation. Okay.
Yeah.
And the gamma asteroid, the damage.
Right. So yeah. So the idea, yes. So this is. So one possibility, right, is when you have RPA-coded single-strand DNA-generated R loop, that is already sufficient to recruit ATR and perhaps activators such as ETA1. So that's enough to give you some ATR activity there to phosphorate RPA. I don't think this is a full activation of the pathway, but you do have some ATR activation. Yeah.
Just to challenge a little bit your model, especially your attempt to get selective killing of with ATM but not ATR, with ATR but not ATM inhibitors. And in the splicing factor mutations, patient-derived mutations, you say it's increased level of R loops, isn't it? And that are actually these cells, if I understood properly, are sensitive to ATR but not to ATM.
But not ATM, yeah.
So how do you explain, because we know that that R loops are actually causing what actually Marco said, a collision with replication fork. And you also see that most probably it's collapse of DNA replication fork and formation double-strand breaks. It would be actually ATM dependent. Right. How can you be sure this is actually so strictly sensitive to ATR and not ATM, not to ATM inhibitors? If we envisage that this actually double-strand breaks formation also must be there, double-strand break formation.
Yeah, you're right. So if you induce R loop, right, induce R loop by expressing this mutant, splicing factor mutant. What we see is we can see phospho RPA, which is ATR dependent, but we never see phospho check 2. So we don't think the problem itself is giving you a lot of DNA breaks. But after you remove ATR, you inhibit ATR, now you get break. So R loop is the initial problem, but when ATR is there, you start to see response, but you don't see the full DNA damage yet. Until you inhibit ATR, now you see a lot of DNA damage. I bet ATM is activated at that time, not the initial phase.
So I'm a bit confused about when you talk about...
Where are you?
Yeah, I'm here.
Okay.
So when you talk about when you do use inhibitors, you increase R loop. And on the other hand, you also say a lot of mutations in the splicing components are heterozygous, which means they are gain of function mutants. So how you explain these things like...
Okay, that's a great point. So gain of function doesn't mean it's gain of positive function. Here it's gain of a negative. It's an interference of normal splicing. It's a dominant mutant. Does it make sense to you? Okay, good. Yeah. Yeah.
I think one of the important function of the checkpoint is to block the mitotic entry. Would you think about that some of the phenotype you saw is actually happening in mitosis? For example, there's a paper published a couple of years ago that did a ATR inhibitor and found that some of the phenotype can be completely suppressed by CDC25 knockout.
Right.
How do you think about that?
Resistant mechanisms you're talking about. Yes, that's a great question. Yeah. So that's definitely something people keep in mind when people develop clinical trials. ATR inhibitor is just like any other targeted therapy. Resistant will be important to issue to investigate. Yes.
I'm just curious that what do you think the normal function of ATR as in terms of regulating R loop formation in the context of splicing? whether you see ATR inhibition would actually affecting splicing. As you say, the splicing mutant cells are sensitive to splicing inhibitors, whether some of the synergistic could be attributed to splicing changes.
Right, right. Well, I mean, when I think about the normal function of ATR, right, so ATR is perhaps there constantly responding to a lot of intrinsic problem, including intrinsic problem arising from splicing defect. That, yeah, perhaps is the way to look at it. Yeah, it's not directly involved in splicing, but when there's something wrong with splicing, ATR is a responder to that. It tries to kind of antagonize the consequence of splicing defect.
Okay, we have last question.
Yes, I have a quick one. You mentioned during the questions that you have our loops formation in G1 and in S phase, right? And perhaps the more frequent ones are in G1, but where do you think your synthetic lethality is happening? Do you think it's cell cycle specific in S phase or not?
Yeah, it's in S phase. Yeah, when we look at DNA damage, it's mostly in replicating cells. Yeah.
Okay, if there is no further question, let's thank Dr. Zhou again.
Thank you.